So welcome everyone to this seminar in the series of transport properties, where we started off by looking at electron transport and atomic transport, ions, bigger molecules, and now bigger systems. This will be held by Professor and our prefect Daniel Söderberg, who is an expert on flow of fiber systems. So please, and we do like we have done before, that you give a lecture, and then depending on how many we are, we do breakout rooms and discuss, and then we will interrogate you, Daniel, on comments and questions. So please, Daniel.
Yes, so thinking about giving a seminar like this, and I thought I had 2 hours, so this will be compressed compared to my original thinking. I chose to talk about mobility and diffusion in crowded fibrillar systems, and I decided to do it also to try to elucidate how we have ended up in this, what we're doing today, actually. And it has a starting point, the beginning. I think it was an intro. It all starts with the beginning, so that's a very simple start. But the beginning was actually that we, as a part of Wallenberg Wood Science Center, the first cohort of PhD students spun kind of strong filaments from nanocellulose. And this was from Carl Håkansson, who was a PhD student of mine. It was really a deep thinking result, I have to say. It took a couple of years to discuss, and Lars Wågberg and Fredrik Lundell were sort of together with me, had an idea of that we should think about how we can align stuff. It was called the Orienteringsgruppen. So the idea was, how can you align things that are very small? It's a mix with 2 PhD students primarily active, then Carl Håkansson and Andreas Fall. Andreas is today at RISE. It's a mix of fiber technology, the colloidal chemistry, hydrodynamics, and some process technology. However, and it's so cool to do this afterwards, because you can rewrite history, because everyone becomes slightly less remember better and better, actually, to be honest. But it's based on the concept developed of the sol-gel transition that is induced by working with pH, or by using salts and highly charged nanocellulose at kind of low concentration. You can go from the liquid-like behavior, which indicates that you have a mobility. So if you have a fibril, as we see to the left, it will move, and if we gel it or lock it, whatever you want to call it, we can get something that behaves as a solid. And this combination was really the starting point for combining with some process knowledge. And what we did was develop a setup where we can push in water and CNF and use something called microfluidics. I know that you have seen it before, so I won't dwell on it, but it's sort of based on this idea coming specifically from Andreas, or the results from Andreas Fall's thesis, and then applying it in a hydrodynamic manner. I want to show this, and I saw that Cornelia is part of it. This is Cornelia picking up one of these filaments that is spun out, and I think that it's kind of in these situations we started to talk about this liquid water, solid water type, and that it's actually more water in this than we have in seawater in this system. And it's still, the filaments can become 2 meters long when they are gelled like this. This is what we think we do. It is a dispersion. It consists of nanofibers. Of course, these are then exaggerated in this image. What happens today, we typically do it in a couple of steps. We have a dispersion. We do a detachment and acceleration with water that aligns a little bit. So it's a system where you control the transport of fibers, but also the orientation. And then you do a second step. You align and you gel it with any type of acid. And then you can spin this fiber. So that was the starting point. you get out something that looks like this. I showed, so this is a fiber. It turns out that then when they are good, they typically are very cylindrical. When they're less good, they are more flat or uneven. I think that actually that has a meaning, which we still don't understand. We can also see that the surface of the filament that is spun has a very characteristic pattern. It is repeated over and over. It appears in other areas as well. There is something called interfacial complexation as a means of making filaments, and they look the same. And I actually think that this surface pattern has a deeper meaning than just being rough. There is something behind this. And of course, what is the knowledge behind this? And then the starting point is that we in this had a mix. We are controlling the transport of fibrils. We think that we know how we align them. We know which process we use to lock them, so we get the gel. So you're going from this non-aligned system to an aligned system that stays aligned because you are changing the electrostatic repulsion. So there's a lot of things happening. There's a lot of transport processes in this, actually, which is interesting at the same time. And we had, in this first publication that Carl made, we had this paragraph outlining in a very scientific manner under which conditions this spinning worked. And it's based on a timescale argument that says that we have to, I need just want to have my, let's see, pointer. Okay, does it work?
Yeah, yes, you see the, not the, yeah, you see a pointer, yeah.
So we have one argument that says that the alignment time, you quickly need to align your fibrils or fibers. And that time needs to be faster than you gel it using the ion that you, or the protons that you put in, which is that sense also needs to be faster than it takes to, by Brownian motion, go back to isotropy. And also that you need to get ions into this system, this filament, in this apparatus, otherwise it's not gelled in the center. So it's a set of timescales that you need to control. And we had some estimates, and they are quite, I think, still they apply, but there are specifically something I will end up in, that is this rotational diffusion due to Brownian rearrangement. This is based on the Doy-Edwards idea of Brownian rearrangement or Brownian diffusion. So looking like just quickly on this, the electrostatic repulsion, I mean, in principle, I actually saw that Lars was here. It is all about understanding what happens. I chose to have this image by Andreas Fall. This is a fibril. It has it's a charged surface. You have an ion cloud around it. This creates the electrostatic repulsion that makes your, at a normal pH, you have a mobile system, a dispersion that is liquid-like. So you can control the rheology by controlling the surface charge or electrostatic screening. I chose actually to have a circular fibril here on purpose, because first of all, whatever you do that is non-circular will be subjected to scrutiny if it's the correct model of fibril. So I ignore that. The other part of this is when you start to look into all of this type of system, it's not only nanocelluloses, it's also in our cells we have fibrillar systems that are quite similar, and also working with carbon nanotubes are similar when it comes to the fibrillar behavior. So yeah, even if we talk about nanocellulose and we know everything about nanocellulose, we think there are reasons to use this for other areas as well. But talking about this of spinning, so we can think about two ways of doing this. The first thing that was published was Andreas Walter, and they took a dispersion and pushed it out in a coagulating bath to the left, and the coagulation bath can be quite many things. What it does, it is of course gels the dispersion into a filament string, and we had something totally different. And it's not so that we want to be complex that we made this flow focusing. It's also about controlling the transport processes. So one of the things that applies for both of them is the first thing is that you need to ensure gelling by having contact with, you need enough with ions or protons, and you need enough time to gel the thread. And that is upheld in this system as well as in both systems by making them sufficiently big. The second part which is then, so I actually say that this is quite straightforward. It's not so much to discuss. There's one thing I should have pointed out. Typically, when we had this discussion and did the calculation in this nice paragraph, we didn't think about that we're consuming the ions that diffuse into the filament. So, and that is a little bit something to think about, because of course, if you have a system and you have ions transported in, they will in some way be consumed while they are going into the system. So it's slower than we think, the gelling time, or the gelling time is longer, the process is slower. But then we have this alignment perspective, and I think there's something, we come from the hydrodynamic side, and that is, I know that most of you have seen it before. This is the alignment by a flow field by Jeffrey. It's more than 100 years ago. It's an analytic expression how a stiff ellipsoid behaves in a shear field. And then Thomas has made this simulation to the right. So we're now looking how these ellipsoids behave when we shear the system. One thing here that I want to bring up is it definitely, the simulation to the right and the model assumes that it's a non-interacting system. So it's dilute. And that should be kept in mind. And we can also see to the right that yes, it's moving, it aligns, but you can also see from time to time that there seems to be a little bit of motion that is not only alignment. And I will come back to that, because that's a knowledge that a lot of people miss out on. So taking that knowledge and thinking about the why, how do we apply this with alignment in these two systems, I want to bring up one thing to think about. And I don't know how many, I shouldn't brag, but I get a lot of since I'm stupid, I get a lot of papers to be referee of, and say yes to them, and then you read them. And the concept of using shear for alignment, because you have published on that, it's everywhere. In what I get, I mean, of course you get something that you're supposed to be expert on, and the shear, we're using shear to get alignment. It's used as a general self-explaining thing that explains everything, and it's used widely and erroneously at many times. And I want to point out why this is wrong. So if we take the leftmost system, you could argue that the syringe is also creating shear and aligns exactly as the flow focusing. So if we then just make a simple simulation, looking at the flow field of something coming out of a syringe, so you have the tip of the syringe here, inside you have a flow field, and we have the velocity profile here. What we have is it's a bath around it. And we can also see that if we then calculate the maximum shear, we see that shear occurs at the surfaces, at the walls. In the center, there is no shear. So yes, if we want to align everything, it won't happen here, because in the center, there is no shear. It's only shear at the walls. It's even, and this is I have shown this before for some of you, this is an interesting publication that passed by, which really shows that you misunderstand this. It's published, so it should be at some point quality control. This is a velocity profile that allegedly makes alignment in the center. Then they have plotted that the shear grade looks like this, which is of course not true, because if you have a velocity profile that is flat, there is no shear. It actually looks like this, the correct. So yes, you shear a system that looks like a syringe, but you only shear at the walls. So what really happens in the syringe? Again, this is the same simulation in the center here as you saw before. It's a shear field. We will see that we start to run the system. You align fibrils. But if you look at it from a 3D perspective, so you also look, you take this and then you look from the top down, it looks like to the right. So this movie illustrates the leftmost volume is ellipsoids in a shear flow, like on the walls on a syringe. And then we have the two projections, one from the side and one from the top. And yes, from the side, the knowledge that everything is aligned, it seems to work. From the top, it's not really super aligned, honestly. There's another version of that, and that is using something called elongational flow. And I will come back to that a little bit. That is what we do in the flow focusing. If we then have the same type of simulation, it looks like this. It's kind of better aligned in all directions. And this is, I mean, this is no news. It's kind of well known. I put this in because I think it's beautiful. I did it for two reasons. This is feeling like we're working with Nobel Prize concepts. So this is a publication in Science 25 years ago, looking at what happens to polymers in a shear flow. And Stephen Shu, which then got the Nobel Prize, not for that, actually. He did it because he invented a method to cool and trap atoms with laser light, but still, it's a Nobel Prize winner. Have studied this, and he in this paper they clearly do exactly. If you look at the mathematics, if you have a shear that is this everyone thinks aligns, it actually contains two parts. One part of the shear field is elongation, that you're stretching, and the other part is that you're rotating. And this actually happens then if you have flexible polymers. So here they have done an experiment, and what we can see here are different polymer at different time steps passing, looking from the top through a channel where you have shear. So the flow is in this direction, and you can see that you are changing between something that is globular to something that is stretched. And then you can compare that for different shear, different type of flow fields. And then they get this, that the mean fractional extension, so the average extension of the polymer, given the flow properties, it's clearly different, that the shear flow does not give at all the same result as the elongational flow, because shear flow is a composition of rotation and elongation. So what we do actually in the flow focusing is an elongational flow. We have a core fluid that is accelerated by the flow focusing liquids from the sides. In the center, we can see that we have, in principle, a pure acceleration. There will be shear in these systems, but it's on the walls where we only have liquid. So where we have our fibers that we want to orient, there is no shear. And therefore, you can say that no, there's no shear in this system, but it actually is really, really good at aligning. And it only aligns then because we have elongational flow in the center. And this we have characterized, for example, using Sachs measurements and DSU, so where you can look at this on the nanoscale. And on the top of this, we see that nanocellulose is birefringent. So if you do this type of setup, this is done by having a colored core flow. So here you have a dye. But if you do this with polarized light, you see that the CNF or the nanocellulose is birefringent. So it actually helps of polarizing light. But based on that, we have this. The idea that we had is that if we ensure that we have alignment and we make sure also that we freeze this alignment as quickly as possible before Brownian motion will come into play and change the alignment, because on nanoscale, Brownian motion is quite strong, this will work. So we are promoting alignment, but we have this de-alignment by rotary diffusion. And this is a beautiful slide made by, I think, made by Thomas. It is it's nice because we have Jeffrey, that's Jeffrey orbit, and Mr. Brownian motion. So what we are doing is fighting between Mr. Jeffrey and Mr. Brown. Alignment is what we want, and Mr. Brown is the thermal fluctuations at the molecular level. And then it turns out, and I just want to show that having this setup that we use to spin, Thomas developed this with us. Took a second step before it sort of disappeared, is the wrong word, but went to Stony Brook. So we use the same setup as for spinning with polarized light. So we have a laser and two polarized 90-degree shifted polarized films. We have a system where we can pump, and when we have quite expensive three-valve switch, three valves that can switch instantly to stop the flow. We can do something called flow stop. And when you observe this with polarized light, you can actually see where you have high intensity, you have high alignment. And this is how it looks, and it's also worked by Christoph, who was a postdoc when Thomas disappeared. And Thomas worked on this continuously at Stony Brook. But what we have is we have a system where it's flowing, the high alignment, because of the birefringence of cellulose, which is highly aligned, we get a lot of light through. And at a certain time, we stop the flow, and then it de-aligns. So we're transporting the fibers, we're affecting the alignment. As quickly as we stop, another process switches in, Brownian motion, which is both true and maybe not fully the truth, because of course when we run this system, so the square here is the intensity that is plotted as a function of time. So when we stop, we get a decay. There is of course always a continuous decay because the Brownian motion is not switched on. It's just that alignment is stronger than the Brownian motion. And Christoph did something that was quite interesting, because when you do dynamic light scattering, this is in principle what you do. You have a system where you assume that you have spherical particles that are non-interacting and non-sedimenting. You can apply this similarly to what we did, and then you can actually run the system. You can take the decay curve that you get out of this, and you can use some clever mathematics, and you can estimate the length distribution of your fibrillar system. And this is really an estimate, and we could ask Thomas how many discussions he had with reviewers about this, how to fit things into this decay curve. It's an interesting thing. But in principle, we today talk about that we can estimate an hydrodynamic length. So that's exactly similar to the hydrodynamic radius in DLS. It estimates how the system behaves and gives a characteristic length that you can couple to the behavior. That was taken further specifically by Krishna and Fredrik, and they sort of implemented this and made some simulations. So we have made a quite good publication that we typically today try to separate, and I won't go into the details. You can do the virtual reality thing. This is a digital twin, but in principle, we typically try to separate by fast things that we call short things, and short hydrodynamic lengths, and things that rotate slower that are then long hydrodynamic lengths. All of this assumes one thing, and that is then that we have a dilute system. And is that really true? I think that the reason why we actually succeed in spinning is one step further, and that has something that we have spent quite some time in the last years. It's also coupled to this observation, and this is a fantastic experiment by a very well-trained experimentalist. It is me who went into the lab in the morning without asking, taking these 2 jars of CNF as prepared without asking. It's a tempo CNF I think it's 820 micromolars per gram. It's non-gelled, so it's pH 7 about-ish. There is no lab book for this. It's just I want to know what happens, so I took it and I just sabotaged the sample. But you shake it violently, and then you see, of course, that you get different. You get some sort of buoyancy, but you can also see that bubbles stick. And this is 2 concentrations. If this was a dilute system, I could never explain this, of course. This means that there is an interaction in the system. There is an interacting network, and that has been, I mean, this is not novel, and it's widely exploited, and we see a couple of theses recently from the department coming out of WWSC that is really looking into this and making use of it in various ways. So what controls the alignment, the rotary diffusion is then that comes up? Is it that we can say that it's Brownian and that it's dilute? You can use this, it's called Doy-Edwards assumption. Maybe not. We did quite some rheology with this. This is a little bit messy curve, but I would like to point out that this is then, it's a stable dispersion, looking at the effect of concentration, looking at primarily we're here interested actually in the storage modulus, so that's the elasticity. If you have a network, you have elastic system. A liquid water doesn't have any elasticity, it only has viscosity. And what we found out was that this is a completely complicated thing, and you get in a lot of debate with the reviewers about what happens, because rheology is difficult. But in principle, what we could repeatedly do is that for low concentrations, with low energy, low frequency, we have an elastic behavior that can switch to a total liquid behavior, whilst if we go up in concentration, it sort of remains as a elastic behavior. So there's a network strength in this, even though it's not gelled. And this is also showing about the same thing, that we can see that if we run the system at a certain shear rate, and then increase the shear rate. So what we really do is we are thinking, so this is actually 0.303 weight percent, which is the red one here. And then we're jumping between 20 and 5. So we're on this side of this and that side of that. And we can repeatedly go back between sort of a liquid-like behavior to a more network-like behavior. And there is actually some, not much, but there is an impact on the loss modulus, but that's not too strong. So the answer to this is that it's a crowded system that we need to consider. And that was, this is part of that rheology thing as well. We have tried to understand this based on the concept of looking at this as fibers from the perspective of our knowledge for paper making. And one thing that came out to be very important is to try to characterize the system. And this is difficult because it's made by hand. It's definitely not objective. But you can actually start to take different charges. You get different length distributions, and you have about the diameter is about constant on average for these. And then you can start to understand what happens. And we directly applied, and this is not novel at all. This was also used by Andreas Fall in his thesis, but using the concept of the crowding factor, which is actually tells you how many fibers you have in a volume that is the diameter of the length of the fiber. So it's the crowdedness of the system. Assuming that you have stiff isotropic fibers, you can also start to look at how many contact points you have between fibers, just mathematically. And what we end up is that we actually have something that we call a loose network that has elastic energy stored, and that it reduces the alignment by Brownian diffusion. And we are using the fact that CNF is semi-flexible. It is not stiff. For example, for our case, when we do spinning, typically this crowding factor is about 30, and there is about 1 contact point per fiber. I just want to define what a contact point is. These are 4 crossings and 8 contact points. So when it's one contact point, that means that each fiber is in contact with another fiber. I just put it there because at some point Thomas Larsson was confused. So if you do this calculation for the jars I have down here, we can see that these two concentrations, we have about 2 contact points per fiber theoretically in the system. And that actually tells us that it looks like this. This means that to keep something locked in a system, you need 3 contact points. If you want to have a system with stiff rods and have a percolating system, you need 3 contact points in a 3-dimensional world. We only have 1 to 2. But if you have flexible fibers, it becomes different. And this is done by Bill Sampson from the University of Manchester. They have taken a kraft pulp and dyed 10% of the kraft pulp fibers black. They are about 20 micrometers in diameter. They have an average fiber length of 2.5 millimeter, and it's a 0.1% concentration. And this is also about, in this case, about 1 to 2 contact points. But clearly, this is stuck. There's a lot of contact points. And the answer to this is that since you have a system of flexible particles, they can bend into place, and they will bend into place because you're processing your system when you make the fibrils, you pump it, whatever you do. So if these fibers were stiff of the same dimension with the same concentration, they will not form this network. They will actually sediment down to the bottom and disappear. So this is what we try to see. This is Brownian motion of single fibers is, of course, something that is different from Brownian motion from a lot of fibers that is interacting. So we have recently gotten deeper into this with crowded polymer systems. This has also a very, wow, there's a Nobel Prize winner again. So this is something that has been a lot of work done on, and from a lot of theoretical, almost more theoretical than experimental part, discussing what happens to polymers in crowded systems. And it's called reptation, the behavior of a polymer. And reptation comes actually from that the polymer when moving looks like a reptile, as a worm, a snake, worm-like behavior. So reptation for rigid, semi-flexible, and flexible polymers are different. This is what we know. I mean, these are stiff. These stiff would follow what Doig-Edwards derived for non-interacting systems. And there is an ongoing discussion regarding how the stiffness of the polymer affects the crowded systems. And this is about that in the conventional polymer rotation model, the time scale for a chain, if you have this, you have a lot of polymers. The time scale for a chain for sort of moving through a tube that is around is much shorter than the time scale for that the network changes. So it's actually neglecting the effects of entanglement caused by particle shape and stiffness. And Ogic proposed that even slightly flexibility increases the rotational diffusion of fibers, and got a model for that, which is then in contradiction to what Doig's argument is, how you can scale the rotary diffusion DR. There was a publication not too long ago, 2010, where they actually looked at carbon nanotubes in a static agarose gel network. So the network wasn't moving. That actually gave the same result at Ogic. So tubes in both of these systems are always assumed to be static in the time frame from your polymer or fibril moving through the tube, or actually moving a length similar to the length of the fiber. So what we have done, and this is Ahmad's first paper, looking at trying to understand nanofibers and the liquid solvent using a bead spring chain model with interactions set up. It's a mechanistic approach. It's using coarse graining. You can also use, in this system, you can use lattice-Boltzmann's fluids. So you can actually include hydrodynamic interactions, which I think is important. It's not only the molecular interactions. And then he gets this type of behavior. This is a crowded system. We see the beads here, and they stick together. And we can also see this is the time history of one filament moving in a network. So around this filament, there's a lot of other fibers, but you can follow how does the individual fibers move. And I said, why do I say we? Yes, Ahmad's had done a lot of simulations on this, and we have done use sort of different lengths, widths, persistence lengths. So stiffness and persistent lengths are clearly coupled. And at a given number of fibers per unit volume, you can estimate the distance between the fibers with something called the geometric mesh size. And by doing this calculation, you can form this type of diagram where this cursive L it's proportional to the inverse of the stiffness of the nanofibers. And that you have your rotary diffusion as simulated compared to the rotary diffusion predicted by Ojic's expression. And this means that Ojic's expression would be just a straight line, and we can see it here with the dashed lines. The static system is the experiments that were made by Fakhi 2010, and it seems to follow for when you start to come up at a certain L, you start to follow it directly. We can see that you have the Doi-Edwards relation, and by implementing the fact that you have semi-flexibility, you are getting a totally new relation, which means that you can't predict the behavior just based on Ojic's Doi's relation. And what it actually says is that you have a totally different model or determining how your fibrils behave, and you have to implement that when you want to design your spinning equipment, for example. And actually, I would say that this is one reason why the spinning works, because if it was only Doi's relation, I don't think that the gelling wouldn't be fast enough. So we actually, on top of what we made before, I would say that the argument today is that this loose network, so the crowded system, is needed to prevent the alignment. It, of course, also obstructs the alignment because it has elasticity. So you need a sufficiently long and strong elongational flow to get alignment. But then this networking crowdedness prevents the de-alignment. This is now looked at as a next step also for controlling diffusion. So this is also from what has been made, and we had one publication with Thomas in Physical Review E, where we have sort of looked at diffusion of nanoparticles in cellulose fibrillar systems, and specifically the latest one that is, I would please have a look at Ahmad's thesis defense. It's the 13th of June. Under preparation for submission is not true, actually, because it has come back and it needs, it's not rejected, at least. It needs some changes. But in principle, what we have done is that you can look at what happens to a particle now in this fibrillar system, depending on the charge between the particles and the fibrillar system. And you can also talk about the size. Here, the size is smaller than the mean distance. It's about the same as the mean distance, and it's larger than the mean distance. Then the simulation starts to look like this. Then you have your particles in the system. You can see here that in this case, it actually kind of kisses and interacts with the fibrils that are moving. In other cases, when you have opposite charge, you can also see that this is how the particle experiences its surroundings. There are fibers here, really, but here we are sort of plotted the virtual walls that the particle sees that prevents it from moving. So this shows that you have a reconfiguring transport system inside this fibrillar network where the particles can move. And this is another way of doing it, looking at having different charge nanoparticles in a fibrillar system and dropping them at a certain time in the center, and you can see how you can control the spreading rate of them depending on the particle the charge. It's a very mechanistic approach, I have to say, but it has a possibility to include the electrostatics as well. And with that, I have almost exactly had my 45 minutes, Mikael.
Thank you very much. Really on time. So we are now, thanks a lot for a very, very interesting and illustrative talk. So Linda, if you could perhaps divide us into some